Good morning, everybody. So, we are learning bacterial genetics. And last class, you have learned a lot of bacterial genetics. And today, I will tell just gene transfer by bacteriophage, that is transduction, and lysogenic conversion and conjugation. But before that, a bit recapitulation. You know, you know uh, what is gene? It is part of DNA which carries the hereditary component from generation to generation. Now, and we are learning bacterial genetics, not human genetics. And of bacteria, one circular chromosome. It is circular, end to end block, circular chromosome. And it is made of double stranded DNA. Double stranded DNA, and these are extra chromosomal DNA, that is called plasmid and transposon. Transposon, these are also free DNA, and these can jump from one plasmid to another, one plasmid to chromosome, from one side of chromosome to another side. For this nature, it is called jumping DNA. Okay, and what is its extra character? It is not self-replicating. Plasmid is self-replicating. It not depend on the replication of chromosome, but this transposon when attached to the plasmid, then it replicates along with the plasmid. When it is attached to the chromosome, it is replicated by the associated with chromosome. And what are the properties? It can carry the drug resistance property from one bacteria to another. And it is of two different sizes. One is very small, 1 to 2 kb, that is called insertion sequence. When size is small, 1 to 2 kb, then it is called insertion sequence. When it is large, it may be from 4 to 25 kb. 4 to 25 kb, then it can help transfer of the drug resistance property, etc. Now, just for recapitulation, this is the DNA structure chromosome, which is made up of two backbone, that is nucleotide, which is composed of one sugar, and this is phosphate. It is sugar, phosphate, alternately arranged. These are two complementary strands. One is complementary to other. It is a helical structure discovered by Watson and Crick. Last bench, yellow sweater. You. ক্যামেরার পাশে শুনছো কি? Yes. And there are bases, purine and pyrimidine. Purine, what are the names? Adenine and guanine. And pyrimidine, from this side, cytosine and thymine. And here attached So, here is one purine, that is adenine, and there is one pyrimidine, thymine, and then if there is T, there will be A. If there is C, there will be G. So, combination is ATGC, always ATGC. And the ratio between this ATCC and AT and GC, it indicates different species of bacteria. Okay. So, ratio of this AT and GC indicates different species of bacteria, as everyone has A and T, G and C, but this ratio differentiates from one bacteria to another. And these are bonded by hydrogen. GC bond is triple layered and it is more stable. So, the GC indicates the genetic code maximally. So, this much you know, and replication you also know. You know, just the recapitulation. When there is replication, there is separation of the strands, you know. So, just like a fork, separation. And when there is separation, RNA is formed. So, central dogma is from DNA, RNA is formed, from RNA, polypeptide or protein is synthesis. This is the ultimatum. That is, central dogma is from DNA, RNA is transcribed, and from RNA, translation of the polypeptides, which will form the different proteins of our different organs. Now, if there is separation, so what will be done? Formation of mRNA, messenger RNA. If it is A, there will be T. If it is G, there will be C. If it is C, there will be ATGC. So, complementary strand is produced in RNA. So, the information coding is passed to the RNA. Now, you know, this RNA comes in the cytoplasm, and by the help of transfer RNA, it goes to the ribosome and then synthesis of the polypeptide. Okay. The terminology is separate: transcription of the messenger RNA and translation of the protein. This much you know what is basic thing before starting for transfer of genetic material. And transfer of genetic material, you know, the methods are what are the names of gene transfer? Okay. So, gene transfer, first transformation, transduction, lysogenic conversion, conjugation. So, transformation you already know. What is transformation? It is the transfer of genetic information from one bacteria to other by the help of naked DNA, that is transposon or plasmid. So, transfer of genetic information from one bacteria to other by the help of naked genes, that means not chromosomal genes. Naked genes are free genes, that means plasmid, transcription, etc. And you know the experiment done by, what is the name? Griffith. He experimented on mice by the help of capsulated pneumococcus. You know, capsulated pneumococcus. That means from one is rough strain and one is rough living. This is rough living but without capsule. So it is rough, and this is smooth having capsule but it is heat killed. At a dead, at a living, and it has virulence factor in the capsule. Virulence factor is in the capsule, but it is dead. It is living, but it has no virulence particle, that is, it has no capsule. So if it is injected to the mice, mice will remain alive. This was the experiment. When he injected this dead one, mice was alive, as there is no virulence factor. But Griffith done mixture of these two, one dead having virulence factor, one is living without virulence factor. Then he injected it to the mice. What was the result? It was seen mice was dead. Why? There must be some genetic code which form capsule in the living rough one. So as it is getting capsule and capsule is virulent and now it is living, so it killed the mice. So how it was possible? There must be some genetic code which passed from this dead living to dead, which helped formation of the virulence factor. So there must be some information. It was proved that it was by the help of free DNA. So there was capsule formation and mice was dead. This was transformation. Now my topic is transduction. Transduction means transfer of genetic material from one bacteria to other by the help of bacteriophage. What is bacteriophage? You know, that is a kind of virus which parasitize one bacteria. ভাইরাস নিজে রেপ্লিকেট করতে পারে না, যখন কোনো হোস্ট সেলের ভেতরে যায় তখন হোস্ট সেলের মেটাবলিক অ্যাপারেটাস ইট স্ন্যাচেস অ্যান্ড ইউজড ফর ইটসেলফ। তাই একটা অলওয়েজ লিভিং সেল দরকার ফর দ্য রেপ্লিকেশন অফ দ্য ভাইরাস। ব্যাকটেরিয়া কিন্তু বাইরে বাঁচতে পারে, ভাইরাস বাঁচতে পারে না, মাল্টিপ্লাই করতে পারে না। সো হেয়ার ব্যাকটেরিওফাজ এন্টারিং ওয়ান ব্যাকটেরিয়া অ্যান্ড destroys the genetic materials of the bacteria and generate its own genetic material. So what is the picture? তোমরা কি পেছন থেকে সবাই দেখতে পাচ্ছ? লাস্ট যারা দেখতে পাচ্ছ কি ছবি? এই জন্যই তো সামনে আসতে বলি। সামনে তো বসার জন্যই তোমাদের সব বানানো হচ্ছে, তোমরা এই দূরে যাচ্ছ। পেছনে যাই না, দেখতে পাচ্ছ কিনা? This is one bacteria having its chromosome. Now one bacteriophage is coming with its nucleus, genetic element. Now when it comes in contact of another bacteria, it delivers its genome within the bacteria and multiply it. Its genome multiplies as bacteria has to multiply, has to survive. So there is multiplication of the, just like virus, there is multiplication of the genomes and destruction of the host chromosome. টুকরো টুকরো হয়ে গেছে, destruction of the host chromosome, and at the same time by some proteins there is formation of the packaging which will act as capsid. Now what happens next? So packaging, নিউক্লিয়াসটা প্যাকেট হয়ে গেল, ক্যাপসিডে ঢুকে গেল, সো মাল্টিপল ভাইরাসেস আর ফর্মড অ্যান্ড হিউজ নাম্বার উইল কজ রাপচার অফ দ্য সেল অ্যান্ড bacteriophage will come out. This is lytic process, lysis of the cell. Okay, this is lytic process of bacteriophage. Next, another process I am going to tell, the transduction. What is this? Sometimes it happens, পেছন থেকে আমি জানি না দেখতে পাচ্ছ কিনা, now please see, here bacteriophage is coming. Now its genome is entering into the cell. There is incorporation of the genome of the bacteriophage with the bacterial cell. Now it is called prophage. It is called prophage. Now this is a new gene. The character is totally new. Okay, totally new. Now this bacteria, still it is bacteria having new gene. When there is binary fission, bacteria always multiply by binary fission. When there is binary fission, there will be division of the cell along with the division of the chromosome. So these chromosome will be divided in the same way. That means as a new chromosome, and it will pass through the all next generation, and these bacteriophage may carry some toxic property. So this toxic property may go to the other bacteria by this prophage. This is called lysogenic conversion. So this is lysogenic conversion. There is no lysis, ফেটে গেল না, it is intact, having a new character, incorporated with the nucleus of the genome of the bacteriophage, and it will transfer from generation to generation. And if it carries any property, that is drug resistance or toxicity, it will deliver it to the next bacteria. Example is Conjugation diphtheria. You know, Conjugation diphtheria usually non-toxic, but there are some phage, that is beta phage. It is carrying the toxigenicity and incorporating to the Conjugation diphtheria. Then it becomes toxic. So by the help of beta phage, toxicity is transferred or delivered to the Conjugation diphtheria. So function of this transduction is transfer of toxigenicity or transfer of drug resistance. So what are the functions of transduction? It is Normally, the genetic material which cannot be transferred by itself from one bacteria to other by the help of sex pilus, যে নিজের প্লাজমিডকে ট্রান্সফার করতে পারে না, সেসবগুলো ট্রান্সফার হবে বাই দ্য হেল্প অফ ব্যাকটেরিওফাজ। সো হোয়াট ইজ ট্রান্সডাকশন? দ্যাট ইজ transfer of the genetic element from one bacteria to other by the help of bacteriophage. It is of two types. One is generalized transduction, when any part of DNA can be transferred by any bacteriophage. Why you are laughing? It is not a story which will give you pleasure. I think very bitter taste. Red sweater. So one is generalized. প্রবলেমটা কি হয় জানো, আমাদের অসুবিধা হয় পড়াতে। যদি আমার সামনে কেউ হাসছে, তুমি যদি কাউকে কিছু বলো যে দেখো তোমার সামনে হাসছে, তুমি একটা সিরিয়াস কিছু বলছো, তোমার প্রবলেম হবে, এটাই না? আমার কিছু কি হবে, তোমরা শুনলে না শুনলে কিছু না, না শুনলে অন্তত হাসবেন বা চুপ করে থাকো, ঠিক আছে? আমাদের তো পড়াশোনা করে আসতে হয়, বলতে হয়, ভুলে যাবো তো। আচ্ছা, so two types of transduction. One is generalized, depends when any part of genome is transferred by any bacteriophage. But another is restricted, that means when specific bacteriophage will carry specific gene to particular bacteria, that is which is required for drug resistance property, which is required for toxigenicity. Toxigenicity কোন কোন ব্যাকটেরিয়া এরকম হতে পারে ট্রান্সডাকশন? ওয়ান ইজ আই হ্যাভ টোল্ড কর্নিব্যাকটেরিয়াম ডিপথেরিয়া, নেক্সট কামস SPE, that means streptococcus having pyrogenic toxicity, next Vibrio cholerae, next botulinum toxin. This can be delivered by the help of bacteriophage. Next comes conjugation. So transduction I have told, lysogenic conversion, you know. Now conjugation. This is one bacteria having chromosome and plasmid. This is another bacteria having chromosome, no plasmid. This is which have plasmid, that is called F plus. F means fertility factor, that it has plasmid which codes for formation of pilus, sex pilus. Pilus means fimbria, but special type of fimbria is called sex pilus, which has sex pilus. They can transfer this plasmid to another bacteria. So who has this plasmid, that is F plus, fertility factor, and who has not this pilus, that is called F minus. So this is donor, this is recipient or female, and this is donor or male. Now, as it is coded for the formation of the pilus, from one pilus, through the pilus, genetic material will pass to the next recipient or female F minus bacteria, then it will gain the genetic material. Okay. What happens? First, one strand is going. So there is missing of one strand, but at the same time there will be formation of the complementary strand, and here one strand is passed, there will be also formation of the complementary strand. Now this is also like this. So it is now donor, as it has the same character of plasmid which can produce the sex pilus for the next bacteria. So now it is donor. Previously without plasmid it was recipient, female, F minus. Now it has capacity to transfer this genetic material. So it is now male, and who has no this character, it is F minus and female. Now it is male. Okay, this is you have to keep in mind. So it is F plus, it is F minus, now it is F plus, now it has capacity to make other bacteria F plus. What is the utility? এত বলার মানে কি? That it can transfer the drug resistance property from one bacteria to another. It has drug resistance property. আমি যদি কোনো একটা পেশেন্টকে অ্যাম্পিসিলিন দিই, তার হয়তো ইকোলাই ইনফেকশন আছে, ইকোলাই ইনফেকশন আছে, but that E. coli has plasmid which is coded for drug resistance to ampicillin. তাহলে ampicillin তখন সেটা কি করে, as we always want to survive, bacteria also wants to survive, সে বাঁচার জন্য কি করবে, তার আশেপাশে যত ব্যাকটেরিয়া আছে, ইকোলাই আছে, সবাইকে সেটা ট্রান্সফার করে দেবে, সবাইকে মেল করে দেবে, সবাইকে ডোনার করে দেবে, যাতে ইমিডিয়েটলি সব ব্যাকটেরিয়ার মধ্যে এই ড্রাগ রেজিস্ট্যান্স প্রপার্টিটা আসে, তবে সে প্রোটেস্ট করতে পারবে, তার আর ইনফেকশন হবে না, ব্যাকটেরিয়াটা মরবে না, হ্যাঁ, পেশেন্ট সাফার করবে কিন্তু ব্যাকটেরিয়াটা মরবে না। সো দিস ইজ দ্য ড্রাগ রেজিস্ট্যান্স প্রপার্টি টু স্প্রেড টু দ্য আদার bacteria. Bacteria is trying to make all the females to convert into male to supply the drug resistance property via the plasmid. এটাই হচ্ছে conjugation. Conjugation from one bacteria to other by the help of sex pilus, transfer of the genetic material. Okay. Now example of conjugation. Conjugation আরেকটা প্রবলেম কি, this extra chromosomal element what is the danger যেটা আমরা ফেস করতে যাচ্ছি, দ্যাট ইজ মাল্টি ড্রাগ রেজিস্ট্যান্সি। আমাদের এমন একটা সময় আসছে আমরা সব ড্রাগেই মোটামুটি রেজিস্ট্যান্ট হয়ে যাচ্ছি, এরপর এমন ড্রাগ থাকবে না যা দিলে আমার কিওর হব আমি। তুমি দেখবে অনেকেই ডক্টর চেঞ্জ করছেন, কেন, তার রোগ সারছে না। আসলে তা না, আসলে তার ড্রাগ রেজিস্ট্যান্সি ডেভেলপ করে গেছে এগেনস্ট মাল্টিপল ড্রাগস। কেন? হ্যাপোজার্ড ইউজ অফ অ্যান্টিবায়োটিক্স, আমরা এমন মুড়ির মতো অ্যান্টিবায়োটিক খাই একটুতে, আমরা ইমিউনিটির উপর ডিপেন্ড করি না, তাই ব্যাকটেরিয়া সার্ভাইভ করার জন্য তার রেজিস্ট্যান্স তৈরি করে নিচ্ছে। বিভিন্ন ড্রাগ, সেফালেক্সিন, অ্যাম্পিসিলিন, ইমিপেনেম, মেরোপেনেম অ্যান্ড ডক্সিসাইক্লিন, সব ড্রাগের এগেনস্টে অনেক দিন ধরে ইউজ করা হচ্ছে, অনেক দিন ধরে ব্যাকটেরিয়া ড্রাগগুলো ফেস করছে। ব্যাকটেরিয়া বাই দ্য হেল্প অফ মিউটেশন চেঞ্জিং দ্য জেনেটিক ক্যারেক্টার টু রেজিস্ট দ্য অ্যান্টিবায়োটিক। এখন ব্যাপারটা কি, এই যে প্লাজমিড, ডেঞ্জারটা কোথায়? ক্রোমোজোমের যেটা ডেঞ্জার হচ্ছে প্লাজমিড, অ্যাজ ইট ক্যারিজ দ্য ড্রাগ রেজিস্ট্যান্স প্রপার্টিজ, it may carry more than 8 drug resistant against 8 antibiotics, মানে একটা ব্যাকটেরিয়া 8টা ড্রাগের এগেনস্টেও রেজিস্ট্যান্স প্রপার্টি শো করতে পারে, as there is genetic code within the plasmid. So this is suppose genetic code for ampicillin, this is for meropenem, this is for imipenem, this is for cephalexin. So এতগুলো ড্রাগ সে যখন আরেকটায় ট্রান্সফার করবে, সেই ব্যাকটেরিয়াটাও এতগুলো ড্রাগে রেজিস্ট্যান্ট হয়ে যাবে। ওকে, নাও দেয়ার আর টু ফ্যাক্টরস, ওয়ান ইজ RTF, resistance transfer factor. This is the plasmid, রেজিস্ট্যান্স কে ট্রান্সফার করতে পারে। কতগুলো রেজিস্ট্যান্স? Those are called small r, resistant determinant. তাহলে স্মল আর এগুলো হচ্ছে রেজিস্ট্যান্ট ডিটারমিনেন্ট, আর পুরো জিনিসটাকে ট্রান্সফার করে যে ক্যাপাসিটি, দ্যাট ইজ RTF, রেজিস্ট্যান্স ট্রান্সফার ফ্যাক্টর। ধরুন একটা গরুর গাড়ির চাকার অনেকগুলো স্পোক আছে, সেই স্পোকগুলো সব হচ্ছে ড্রাগ রেজিস্ট্যান্ট প্রপার্টি কোডেড আছে, আর চাকাটাকে কেউ ঘুরিয়ে নিয়ে যাবে তো, তাই তো, দ্যাট ইজ RTF, হলো। দেন ইট ইজ পাসেস টু দ্য নেক্সট ব্যাকটেরিয়া, সেইভাবে সব ব্যাকটেরিয়া আশেপাশে সবাইকে এতগুলো ড্রাগের এগেনস্টে রেজিস্টার করে দেয়। Here is the importance of conjugation, and this is done in the engineering, that is called genetic engineering. Genetic engineering, that is recombinant DNA technology. এখানে কি হয়, প্লাজমিডটাকে বার করে নেওয়া হয়, প্লাজমিডের উপরে কেটে বাই দ্য রেস্ট্রিকশন এন্ডোনিউক্লিয়াস কাঁচির মতো কেটে কেটে ধরো বাজে জিনিসটা ফেলে দেওয়া হলো, এবং হুইচ ইজ সুটেবল দ্যাট ইজ অ্যাডেড, তারপর সেটাকে কোনো ব্যাকটেরিয়ার মধ্যে ইন্ট্রোডিউস করে দেওয়া হলো, সেটাকে মাল্টিপ্লাই যদি করি তাহলে ভালো ক্যারেক্টার নিয়ে ব্যাকটেরিয়াগুলো তৈরি হলো, অনেক আমরা ভালো ক্যারেক্টারের জিন পেলাম, সেটা তারপরে কাউকে ইনজেক্ট করে দেওয়া হয়। সো দিস ইজ দ্য বেস অফ genetic engineering by the help of mainly plasmid, as it is free DNA and it has various capacity. Now I will go for the hello তোমরা সবই আগের দিন দেখেছো। This is the genetic structure, you know, structure of DNA, you know. These are thymine, adenine, guanine, cytosine, everything you know. This is the helical structure, double helix. These are the this is the main thing. This is plasmid. Okay, previously everything you know. This is plasmid. You can see this is the bacterial chromosome, and this is circular plasmid. It is also circular. It is 1 micron, and কতগুলো মাইক্রন থাকে একটা ব্যাকটেরিয়ায়, কতগুলো বেস পেয়ার থাকে? 4000 কেবি, কিলো বেস পেয়ার। হিউম্যানে 3000, তার মানে হিউম্যানের চাইতে ব্যাকটেরিয়াতে বেশি বেস পেয়ার থাকে, ঠিক আছে। হিউম্যানে যেটা 3000, ব্যাকটেরিয়া 4000, 1 কেবি, দ্যাট মিনস 1000 বেস পেয়ার। যাই হোক, নাও it is the plasmid. Plasmids are circular DNA molecule in the cytoplasm of the bacteria, capable of autonomous replication, can transfer genes from one cell to another, act as vector in genetic engineering. So it acts as vector in the genetic engineering, can also present in the yeast. yeastের মধ্যে প্লাজমিড থাকে, তাই এটা কি করে যাবে? ওই যে টেস্ট এটা চলো। Okay. Now we can see blue colored, these are the plasmids. It may be multiple in a cell, not only one, and this is the bacterial chromosome. Plasmids seem to be ubiquitous in bacteria, may encode genetic information for properties. These are the properties of plasmid. Resistance to antibiotics, bacteriocins production. What is bacteriocins? Bacteriocins are antibiotic-like substance produced by the bacteria, and when it spreads around it, the other bacteria may be killed by this bacteriocin. তাই একটা বডির মধ্যে যখন একটা ব্যাকটেরিয়া ব্যাকটেরিওসিন তৈরি করে তখন অন্য ব্যাকটেরিয়া তাতে কিন্তু মারা যেতে পারে, জাস্ট লাইক অ্যান্টিবায়োটিক, দ্যাট ইজ কল্ড ব্যাকটেরিওসিন। Then enterotoxin production, then enhanced pathogenicity, reduced sensitivity to mutagens, degrade complex organic molecules. So mainly resistance to antibiotics and bacteriocins production, exotoxin production. Okay. This is R plasmid. So these are the different sites for drug resistance property. This is the area for chloramphenicol resistance gene. This is the carbonicillin resistance gene. This is tetracycline resistance gene, and this is a pilus synthesis. So plasmid has the code to synthesize the pilus through which it will be transferred. So it is also coded in the plasmid. And this plasmid is called R plasmid. That means which is supplying the resistance property. Next, plasmids can be integrated with the chromosome, and episomes integrated from plasmid with DNA. So this is bacterial DNA, these are the plasmids. When it is integrated, suppose one plasmid is attached to the chromosome, blue color you can see, this is HFR, highly frequency transferable area. It is highly powerful. যেখানে একটা plasmid is incorporated with the plasmid DNA incorporated with the chromosomal DNA. This area is highly active. So HFR. Same picture you can see. Plasmids can be transmissible and not transmissible, it may so happen. And this is the mobile plasmid, this is the sex pilus, and this bacteria has no plasmid. Okay, now it is donor and it is recipient, and from this by the sex pilus there is connection and plasmid is passing. You can see one strand is going. Okay, one strand is going, and against this strand complementary strand will be produced. At the same time there is production of the complementary strands. Now as it was recipient, now it is also new donor, as it has the capacity to transfer by the help of such plasmid. So this is new donor, this is new donor. And one thing, you know, phenotypic expression and genotypic expression. তোমরা জানো phenotypic, that means when there is change or mutation by the influence of environment, that means which is temporary, that is called phenotypic expression. Example is Salmonella typhi. Salmonella typhi has peritrichous flagella. If it is Salmonella typhi, it has multiple flagella, peritrichous flagella. But if it is inoculated in media which contains carbolic acid, okay, then there will be growth of bacteria but without flagella. কোনো flagella থাকবে না. But if I put it in again in the normal media which has no carboxylic acid, again there will be formation of flagella. So this is temporary. This is due to environmental change by the addition of the carboxylic acid. So this is phenotypic change in the bacterial genes, and genotypic change is which is permanent, okay, which is permanent change, and that change, that mutation will be carried from generation to generation. Same thing. Now, principles of genetic variation, genotypic variation, that is change by mutation, phenotypic genotypic variation, change by mutation, and genotypic by transfer of genes. একটা জিন আরেকটা জিনে চলে গিয়ে সেই জিনটাকে চেঞ্জ করে দিল, that is permanent, that is genotypic transfer. What are the examples? Transformation. Transformation, একটা ব্যাকটেরিয়ার জিন ছিল না, আমি আবার একটা জিন দিয়ে দিলাম, ট্রান্সফার করে দিলাম. So transformation, transduction, lysogenic conversion, conjugation, all are genotypic change, and this is permanent. This is also example by template formation. Now mutation, already you know, there are point mutation, frame shift mutation, and there is multiple site mutation. Multiple site, that means at different site of the chain, genome is changed. It may be addition of one base, deletion of one base, substitution of one base, or duplication of one base, that is whole genome, whole chain is changed at multiple site. Example, you can see that is change in this A is shifted. So total genetic chain is changed, as it is GT, G will come, GTG, actually it was AGT, and now CC, A is here. So it is changed and it is permanent. Mutagenic agents, ultraviolet ray, alkylating agents, acridine dye, etc. Now, transduction, you can see transduction, use of bacteriophage, bacterial virus transfer DNA between cells. Transformation, in transformation there is naked DNA is taken up from the environment by bacterial cells. You can see this is the sex pilus, one genetic material is transferring there by the help of conjugation. তোমরা এটা জানো গ্রিফিথেরটা, you know it already. This is, I have already told, that is transduction is transferred as DNA from bacteria, from one bacteria to another by bacteriophage. Here is the bacteriophage, it has own genome is attached here. Now lysis of the bacterial genome, and there is replication of the bacteriophage genome, replication of bacteriophage. Then there is capsid formation, and again multiple bacteriophage are formed. There is lysis of the bacterial cell and it is coming out. So it is lytic cycle. Another process, what is happening from bacteriophage, the new genetic material is incorporated. This is the red portion. Now it is a new gene having another new genetic material from the bacteriophage. Now it may have toxigenic property, it may have drug resistance property, and when it will pass to other bacteria, same property will be transferred. দেখেছো bacteriophage, you know very well. Conjugation experimented by Lederberg and Tatum in Klebsiella. It was experimented in the Klebsiella. Now you can see the picture. This is the fertility factor, that is plasmid. It is by the sex pilus entering another bacteria which has no pilus. So it is female, it is F minus. Now fertility factor plasmid is transferred, it will be again F plus or donor. You can see there is conjugation by transfer. These are the different genes which are responsible for different bacterial resistance. This is the conjugation, same picture you can see. Same picture you can see. Here is the plasmid. This is F plus, as it has the plasmid which coded for formation of sex pilus, and it is F minus. So it is female or recipient, it is donor, and now it is transferring the genetic material. So it is also having the same plasmid. Now it is also F plus, it is also F plus. Same picture. So you can see the lysogenic conversion, that is the first DNA, it is incorporated with the bacterial DNA, and now after binary fission, same character, same new character is transferring to other bacteria by the help of bacteriophage. And here is the formation of the bacteriophage only by the process of lysis. So it is lytic cycle, this is lysogenic cycle or temperate cycle. This is bacteriocin. This is the bacteriocin spreading from the bacteria which will prevent the attack of other bacteria. Same thing. So this is all. Thank you.
Okay, what do you know about, what do you understand? You may even say it in Bengali, but I would prefer it if you say it in English, because in the exam you will have to present your answers in English. But still, if you want, you can say in whatever language you want. বলো কি বুঝেছো acute inflammation মানে কি বুঝেছো? মাইক্রোব একটা অ্যাটাক করলো, দেন there is events hyperemia and chemotaxis and okay. So how long does it take for an acute inflammation to resolve? Approximately? It develops in about 1 day, instantly also. What? Okay, sit down. Okay, do you know what is chronic inflammation? Okay, if the acute inflammation does not resolve and it continues, then it can be called a chronic inflammation. So these are the questions. Now what I usually do is I will ask you a set of questions and I will see how much you know. Just an assessment of how much you know. And at the end of the class, I will again ask these questions. This is what I have done the previous year also. So I've already asked the first question, what is chronic inflammation? And also these questions that I've marked, these are the frequently asked questions. Mane exam e ashe, right? So which are the cells involved in chronic inflammation? And lymphocytes. Okay. Examples? Example of chronic inflammation? There is one prototypical example which every Indian must know, which is that cholecystitis, yes. More popular? Yes, more popular. Yes. Any long-term disease you name is actually a chronic inflammation. But what is the prototypical example? Tuberculosis? Yeah. And do you know the difference between granulation tissue and granuloma? Please stand up and tell me your name. First, tell me your name. Okay. So granulation tissue formation is not an example of chronic inflammation. So the understanding of that is both are examples of chronic inflammation. Granulation tissue is a non-specific kind of chronic inflammation, and granuloma is seen in granulomatous type of chronic inflammation. This is a very frequently asked question, you know. You will see if you go through the exam papers, this question is asked frequently, right? Please sit down. So these are the things I will be teaching today. Now definition. Chronic inflammation is a response of prolonged duration, weeks or months, in which inflammation, tissue injury and attempts at repair coexist in varying combinations. This means that their inflammatory cells will also be present. Along with that, attempts at repair. Which are the cells involved in repair? Fibroblasts. There will be blood vessel formation, fibroblast and laying down of collagen. There will be fibrosis. So you will see some amount of fibrosis, some new vessel formation. There will be multiple chronic inflammatory cells. The chronic inflammatory cells are firstly macrophages. They are the most important chronic inflammatory cells. Now along with macrophages, there will be lymphocytes, right? Both T and B lymphocytes. Along with that, there will be plasma cells, eosinophils, mast cells. Along with that, you can also have neutrophils. So neutrophils are not just limited to acute inflammation. They can be found in chronic inflammation as well, right? So some types of chronic inflammation show a predominance of neutrophils also. So anyway, so these cells will be there. There will be attempts at repair also, as opposed to acute inflammation. In acute inflammation, there is hyperemia, there might be necrosis, and along with that, there will be many neutrophils, right? So the causes. The first cause you've already said, following acute inflammation. So the body was attacked by an organism. Any external source has attacked the body. Now the body is defending against it but unable to resolve it. So it goes into a chronic phase. The example of this is osteomyelitis. Or say there was a pneumonia, the lung was attacked and then the lung could not resolve the attack. So then it goes into formation of lung abscess. So following an acute inflammation, there can be a chronic inflammation following acute inflammation. Apart from that, recurrent attacks of acute inflammation. Now, say, like she said, someone said, cholecystitis. So what happens is the gallbladder is repeatedly attacked by many, say, microorganisms, right? So what happens? The gallbladder goes into a chronic phase of inflammation, chronic cholecystitis. Or a person, say, he is diabetic or immunocompromised and recurrently gets UTI, or very poor hygiene, especially in women. So recurrently gets UTI. So, and then that person may develop pyelonephritis. That is the ureter, the organism travels up the ureter, goes into the kidney. Then they can form pyelonephritis following UTI, right? And this is the most important, chronic inflammation de novo. Now, this is like this organism, the moment it attacks, the body is not able to contain it. The body is not able to eradicate. The body tries, but is unable. The body instinctively knows that no, I cannot eradicate this organism. This is something new for me. So this organism is chronic inflammatory from the beginning. From the beginning, it forms chronic inflammation without per se any acute phase. So that is the prototypical example. Tuberculosis, this is very important. You will also have separate classes, maybe 2 or 3 on tuberculosis later. The other examples now, hypersensitivity diseases. Now, hypersensitivity diseases, now this is a kind of type 4 hypersensitivity reaction. You've not had class on hypersensitivity reaction. There are 4 types. So, this is an example. Chronic inflammation is usually an example of type 4 hypersensitivity reaction. Now, what happens is, in this also there are 3 types. It can be against own tissue. This is what you were talking about. Say rheumatoid arthritis, SLE, multiple sclerosis, all these kinds of Sjogren syndrome, lots of autoinflammatory diseases. So this is the body is attacking its own tissue. The body does not know that this is not foreign. Bodies are, so ultimately it's not helping the body at all. It is detrimental for the body. The person can even die because of this kind of chronic inflammation. The body is attacking its own tissue and giving rise to chronic inflammation. That example is rheumatoid arthritis, SLE, multiple sclerosis. Or there can be unregulated immune response against microbes. Now this is inflammatory bowel disease. Example is Crohn's disease. You know, inflammatory bowel diseases, there are 2 mainly. One is ulcerative colitis, another is Crohn's disease. You were talking about colon, somebody was saying colon, somebody was saying this, right, from that region. So what happens is, we have gut flora, we have normal gut flora, right? Normal commensal bacteria in our gut. So what happens is, now the products of these microbes are attacked for no reason, does not help the body in any way, but there is chronic inflammation. So this is a reason for development of Crohn's disease. So now again the body is attacking its own tissue unnecessarily, right? Another, this is also very common, against environmental substance. Now, this happens, say there are pollens in the environment, maybe seasonal or maybe just because of cold also. What happens is, does not harm the body in any way. This is not a microbe, it's not a pathogen. But the body does not, in some people this happens, the body does not recognize that this is not harming me in any way. Still the body is launching an attack against this. So that again is an example, bronchial asthma. Hyper-responsiveness of the tracheobronchial tree. This is the definition of bronchial asthma. Hyper-responsiveness of the tracheobronchial tree. Unnecessarily, the mane bole na mosha marte kaman dara, that kind of thing. Unnecessarily, the body is attacking that kind of, that foreign substance that has attacked. Or it's just an environmental commensal maybe that is present. Now, the other example is prolonged exposure to potentially toxic agents. That can be endogenous, exogenous. Now, one example of this, say, prolonged exposure affects the lungs. Smoking can be active, can be passive. Smoking. So that is also an example, right? So that can be endogenous, exogenous. Exogenous is silica, asbestos. These are the causes of inflammatory lung diseases, right? They call interstitial lung diseases, they cause fibrosis of the lung. Examples are silicosis, asbestosis. But apart from that, smoking also, smoker's lung, is an example of chronic inflammation. And endogenous, now atherosclerosis, you've heard of atherosclerosis? Now lipids are deposited in the blood vessels and there is chronic inflammatory reaction and atheroma formation and blockage of the blood vessel. Then there is severe blockage of the blood vessel and then ischemia in the following area. It can also cause myocardial ischemia, etc. So atherosclerosis is also actually an example of chronic inflammation, right? Now features. What happens? There is mononuclear cell infiltration. The mononuclear cells are the macrophages and the lymphocytes mainly. Apart from that, mast cells, eosinophils, some neutrophils. Now these will first infiltrate. There will be some chemotaxic agent secretion. There will be interleukin, TNF alpha, interferon gamma. These will be secreted by the Th cells. There will be T helper cells and the macrophages. They will secrete these interleukins and chemotactic factors like protease, elastase, collagenase, lipase, reactive oxygen species, cytokines, nitric oxide, angiogenesis, growth factor. And because of these biologically active substances, there will be tissue destruction. Tissue destruction will cause necrosis. There will also be hypoxia. So in a region of chronic inflammation, there we can find tissue necrosis, tissue destruction, and mononuclear cell infiltration. So once there is necrosis and there will be angiogenic factors will be secreted. So there will be new blood vessel formation. Along with that, there will be some fibroblasts. The macrophage itself will attract some fibroblasts. What is the function of a fibroblast? Collagenization. There will be fibrosis and collagenization. So healing, there will be attempts at healing, right? So this is a granulation tissue actually. You can see these are the small blood vessels. You can see red areas. Can you see? বোঝা যাচ্ছে? There are some red areas. These are actually small blood vessels. And these multiple blue dots, these are the mononuclear cells, right? So there is necrosis, proliferation of small blood vessels and fibroblasts, granulation tissue formation, healing by fibrosis and laying down of collagen. So this is chronic inflammation. This is what you have to write. Now we come to the cells involved in chronic inflammation. Now firstly macrophages. What are they? They are the main, I have already said, macrophages are the main cells. Where are macrophages derived from? Monocytes. Okay. So can you name some specialized macrophages? In and বুঝতে পারছি না microglia in brain and what? Yes, alveolar macrophages in lungs and Langerhans cells are specialized macrophages, but they are not normal. I'm talking about normal. Yes, they are also called sinus histiocytes in lymph nodes. Sinus histiocytes, same. Spleen and lymph nodes have sinus histiocytes, Kupffer cells, and microglia, alveolar macrophages. So these are specialized macrophages which are derived from the yolk sac, and they directly go into these organs and they rest there to protect these organs against any attack, right? Apart from that, the bone marrow produces monocytes. The monocytes remain in the blood for 1 day. Only 1 day is the lifespan of a monocyte. After that, it goes into tissues, and in the tissues, the macrophage can survive for months to years. It just rests there, rests there, rests there. Now, once there is any kind of the T lymphocytes will attract the macrophages, or the macrophages will try to, now they are what? They are professional phagocytes. This means they will try to engulf the pathogen. They will engulf the pathogen and they will try to contain. Also, they will stimulate the T helper cells. This is called acting as antigen presenting cells. What it does is it engulfs the microbe, and an antigen, it brings the antigen to the cell surface and it attracts T helper cells along with another factor. This you will have a class on this. Now this will attract the T helper cell. The T helper cell will now secrete some chemotactic factors and attract more macrophages, and there will be more antigen presenting cells, and the cycle will go on. So there will be lots of macrophages will be recruited and lots of T cells will be recruited. Along with that, B cells and plasma cells will also be recruited. So the main job of this chronic inflammation is done by the macrophages. The macrophages will secrete the chemotactic factors. Along with that, it will attract the T cells and eventually attract B cells and plasma cells. And along with that, there are two kinds of macrophages, right? One of them will attract the T cells. The other will try to lay down collagen and attract fibroblasts. So there are two kinds of macrophages. See, two major macrophage activation. One is classical, which is activated by microbial products like bacterial endotoxin, involved in killing of bacteria. Alternative pathway, these macrophages are involved in healing and tissue repair. Now the second type of macrophages, they are produced a little later, and they are doing the job of healing and tissue repair. So two kinds of macrophages will be found in any chronic inflammatory site. Is it clear? Next, role of lymphocytes. Now the microbes and other environmental antigens activate T lymphocytes and B lymphocytes. These cells amplify and propagate chronic inflammation. This I've already said, that microbes will firstly, they will activate the T lymphocytes and B lymphocytes. These will then secrete the chemotactic factors like interleukin and TNF alpha, interferon gamma. Interferon gamma is very important in attracting macrophages for the classical pathway of macrophage activation. Interferon gamma, you have to remember interferon gamma. So especially for viruses, right? Now these cells amplify and propagate chronic inflammation because if a T lymphocyte is involved in any chronic inflammation, the inflammation will persist for a long time, because it is a cycle of attracting more macrophages and more T lymphocytes. It's like a vicious cycle. It goes on and on. So many T lymphocytes are recruited at the site of the chronic inflammation. So these cells amplify and propagate chronic inflammation. They act hand in hand with macrophages in a cycle where macrophages act as antigen presenting cells to the T lymphocytes, and T lymphocytes release cytokines which recruit and activate macrophages. Now what is happening? Now the T lymphocytes are and the macrophage is presenting the antigen or the microbe to the T lymphocytes. The T lymphocyte is secreting the chemokines, which is again attracting more macrophages. They are acting hand in hand. The macrophages and T lymphocytes are acting hand in hand with each other. Last bench, please stand up. Please stand up. I told you, I understand it's very late, you may be sleepy. Yes, what contain it? T lymphocytes go to that site. Now the T lymphocytes are secreting some chemotactic factors, which is attracting macrophages. Now the macrophage is engulfing that antigen, that bacteria or that virus or that fungus. The macrophage is trying to engulf it. And that is, there is one antigen along with the microbe. The microbe has an antigen. Now it is bringing that antigen to the cell surface. Now it is presenting that antigen to T helper cell. T helper cell, it is also giving a receptor to the T helper cell. So there are two things, that is one antigen and one receptor on the macrophage. Now the T lymphocyte, the T helper cell is recognizing the receptor and then attaching to the antigen. Then it is secreting some chemotactic factors again. These chemotactic factors are attracting more macrophages and more T lymphocytes. This is how chronic inflammation propagates. Is it clear? Now please explain. না যাকে জিজ্ঞেস করলাম এখন বলো, বললাম তো, আবার কি আমার দিকে তাকিয়ে আছো? বাংলায় বলো, হিন্দিতে বলো, যাতে খুশি। আই ডোন্ট নো হোয়াট ইজ ইউর ল্যাঙ্গুয়েজ। তাহলে কি হলো? ওকে, সামওয়ান এলস প্লিজ এক্সপ্লেন। কেউ বলো, তুমি বলো। আমি অ্যাকচুয়ালি ওকে জিজ্ঞেস করলাম, ঠিক আছে বলো 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 বলো। আরো ম্যাক্রোফাজকে অ্যাট্রাক্ট করছে। ম্যাক্রোফাজটা অ্যাক্ট করছে, চলছে, প্রোপাগেট করছে। এখন বোঝা গেল? কি করবে? আনসার লিখবে না? তাহলে বলবে বুঝতে পারিনি, সরি, নাকি হাবি জাবি কিছু একটা লিখে দেবে। তোমাদের আনসারগুলো এরকম হয় পরে, যে কি লিখেছো না, মানে নিজে আমি ভাবছি যে এখন ধরো খাতা টাতা দেখতে হচ্ছে, তো তোমাদের সিনিয়রদের হচ্ছে, সবারই হবে, তোমাদেরও হবে, ইট ডাজন্ট ম্যাটার। মানে আমি বুঝতে পারি না আমি ঠিক জানি না, এটা প্রত্যেকে। এবং আমি দেখছি যে আমি ভাবলাম ঠিক আছে আমি জুনিয়র মানুষ, আমি হয়তো দেখি প্রফেসর যারা বয়স্ক অন্য জায়গা থেকে রিটায়ার করে এসছে, তারাও বলছে বই খুলে দেখতে হচ্ছে দুটো তিনটে, যে কি লিখেছে বুঝতে পারছি না, আমি ঠিক জানি না এ ঠিক জানে, কি লেভেলের গল্প লিখে রেখেছে। তো এরকমই করবে, তাই তো? এক একটা খাতা দেখতে 1.5 ঘন্টা লেগে যায়। কি করবে? বসো, প্লিজ ট্রাই টু লিসেন। সিট ডাউন প্লিজ, থ্যাংক ইউ। Now there are other B cells also. When there are memory B cells involved, especially in any granulomatous inflammation, memory B cells are involved. What are these memory B cells? Some of the B cells get converted into memory B cells. The B cells were also attracted to the site of chronic inflammation. Now some of them will remember that this antigen had attacked me at one point. Now this antigen attacks again. So the memory B cells will now activate all the other cells. So the body has a memory against this kind of antigen. So we know vaccination also works, right? So if there are memory B cells, it is also very important for granulomatous inflammations, right? Activated B lymphocytes and plasma cells are also present in sites of chronic inflammation. What are these? Do you know? Eglo ki? Yeah, immunoglobulins. So, who secretes these immunoglobulins? Yes. So, activated B lymphocytes and plasma cells are also present in sites of chronic inflammation. Now, this is just extra information. Robbins says that this book, or they don't know what is the role of plasma cells and activated these B cells in sites of chronic inflammation. We are yet to find out what is the role, but they are also present in sites of chronic inflammation. In fact, this is extra information you may remember, you may not remember. You will study thyroid in your paper 2, right? Next semester you will study thyroid, you will study rheumatoid arthritis. So in thyroid, there is a kind of thyroiditis called Hashimoto's thyroiditis. You've heard of Hashimoto's thyroiditis? Yes. What happens is there is tertiary lymphoid follicle formation. This is also a kind of chronic inflammation. In the thyroid, there is lymphoid follicle formation. So the thyroid resembles a lymph node, right? So there are lots of B cells and plasma cells over there. We don't know what is their role, whether they are actively attacking the body or actively attacking any virus or anything. We don't know, but they are also present in sites of chronic inflammation. Now other cells, eosinophils. Now what are these? These pink things? Shojato. Eosinophils, right. Eosin mane? Eosin একটা dye, acidic, right. So the color is pink. Eosiner color hoy pink, right, reddish pink. So phil mane? PHIL, phil, loving, yes. So it loves that pink color. So that is why eosinophils. So these are the eosinophils in a site of chronic inflammation. Now they are mainly found in parasitic infection or immune reactions mediated by IgE. Now they have granules. Eosinophils have coarse orangish pink granules, right? Now these granules contain major basic protein. Now this major basic protein is toxic to helminths, right? Parasites are attacked by eosinophils. So eosinophils' main job is attacking the parasites, but can also be detrimental to the host. Example, allergic reaction, asthma. In a person, sometimes you will see clinicians ask to test for IgE. If an person's IgE is raised, we will find that their blood eosinophil is also raised. What happens is the allergic reaction, as I said, that this pathogen is normally not harmful to people, but some people recognize that as a detrimental or a pathogen. Actually, it recognizes that structure, say a pollen or hay or something, as pathogenic, which is not pathogenic to the body. Now then there is hypersensitivity reaction, IgE secretion, and there will be raised eosinophils, or that site will have lots of eosinophils. So eosinophils can also be involved in chronic inflammation. Mast cells are also there, and neutrophils play a role in persistent bacterial infection, like osteomyelitis or acute and chronic inflammation, like smoker's lung. In smoker's lung, actually the antigen goes on. The person has not stopped smoking. So the antigen is constantly present in the body, and there will be neutrophils, that is acute inflammatory cells, along with chronic inflammatory cells. So neutrophils also play a role in chronic inflammation. Now systemic effects of chronic inflammation, we all know this. Fever. What kind of fever? Usually low grade fever. Usually low grade, can be high grade, but usually low grade fever. There will be anemia, leukocytosis. Which type of blood cell will increase? Usually lymphocytes. And there will be raised DSR and amyloidosis. Long standing chronic inflammation gives rise to secondary amyloidosis, right? These are the lymphocytes. Now I've already told you types of chronic inflammation: chronic non-specific inflammation and chronic granulomatous inflammation. Non-specific inflammation means there will be granulation tissue formation, right? And what is granulomatous inflammation? There will be granuloma formation. What is the difference between granulation tissue and granuloma? ও মা কথাটা কিসের সঙ্গে? Tumor, right? So granuloma is previously it could be found that there were some tumor-like some things were found in the body, say in lungs. There are small dots in a macroscopy, gross. বড়টা দেখছো লাংসে ছোট 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 অনেক টিউমারের মতন দেখাচ্ছে. That can be granuloma. Or say in intestine, you find lots of small. You think that maybe these are, what is this? This is a metastatic cancer. The cancer has spread throughout the intestine or throughout the lung. But actually, when you see under the microscope, you don't find any cancer. You find a granuloma. So granuloma are these small tubercles, maybe small raised areas, right? They have certain configuration. That is why previously when it was named, it was called a granuloma. It may not be raised. It may be very microscopic and you will not find it in gross examination. But that is where the word granuloma comes from. And granulation tissue is that one that I had shown a picture of. There is chronic inflammation. Then there are lots of chronic inflammatory cells. Some of the macrophages are now fibroblasts and they have attracted other fibroblasts. And now the fibroblasts, and there are some lots of small blood vessels. Because of angiogenic factors, lots of small blood vessels have formed. And there are lots of small blood vessels, mononuclear cells, and collagenization and fibrosis along with fibroblasts. That is called granulation tissue. That is different. And what is a granuloma? Granuloma forms when, say, now this is like, say, a country is attacked by some terrorists or someone, right? So now the defense mechanism of the country goes to contain that outside attack. Now the defense mechanism is trying to contain, but it is a kind of bomb, right? Bomb now they cannot do anything about it. They don't know how to defuse the bomb. The bomb might burst. So what will you do? Yeah, and you will try to contain so that the bomb does not burst and spread to the whole area. You will try to contain the explosion. So that is what a granuloma is basically trying to do. It has faced something like a Mycobacterium tuberculosis, that bacilli. Now this bacilli, now the body knows that na eta besh strong ache, I cannot do anything about it. Now what will it do? Firstly, acute inflammatory, agei neutrophils have come and they have died. They have died off, or they know that no, the first line of mechanism won't work. The body knows. Second line, T cells. T cells have come. T cells are seeing that na, amio parbo na, okay, I can't do anything. Now macrophages have come. Now the macrophage is very intelligent. It is trying to eat up. Lots of macrophages have come. Still they cannot do anything about it. What does it do? It forms a granuloma. In the center, there are lots of bacilli which are latent. They are not active. They are not harming the rest of the body. We all know if we are born in India, it is endemic for tuberculosis. All of us within the first year of our lives will be affected by tuberculosis if we are born in India, right? That is why BCG is given at birth. BCG vaccine is given at birth, so that it prevents miliary tuberculosis, right? It prevents widespread tuberculosis. So now the macrophage has come, and the macrophage is now converting itself to epithelioid cells. It looks like epithelium. You've studied epithelium in anatomy. It is the covering. So it looks like epithelium, that is why epithelioid. So So the macrophage is now converting to epithelioid cells. So there are lots of macrophages. Along with that, T cells are also there. And in long standing, if it has been there for a long time, there will be fibrosis surrounding the granuloma. So this is a granuloma. So how is it different from a granulation tissue? Quite different, very different. Only the name seems similar, right? It's quite different. So can anyone please tell me the difference between granulation tissue and granuloma? কে বলবে? পেছনে নীল জামা, তুমি পেছনে তাকাচ্ছ কেন? বলো। তোমরা কি এমনি আসো নাকি? মানে শুনবে না। তুমি বলো হলুদ জামা, তোমার নাম বলো প্লিজ।
Macrophages aggregate systems to form an epithelioid cell and then it contain the
Microbe.
in a particular location so that it cannot spread.
And there are what other cells are there in a granuloma? T cells.
T cells are there, macrophages are there, and
Ring of fibrosis. And in granulation tissue? Granulation tissue, there are lots of mononuclear cells along with small blood vessels, fibroblasts, fibrosis and collagenization. That is granulation tissue formation. Please sit down. So basically any ulcer, if the body has any ulcer, say what is an ulcer? Breach of epithelium, breach of covering, diabetic foot ulcer. So there is granulation tissue formation. What happens is we frequently get samples from say surgical sites, surgical wound sites. Say the surgical wound, the incision is not healing, non-healing ulcer for a long time. We frequently get these samples and we will see chronic non-specific inflammation and granulation tissue formation. This is the report every time, that there is chronic inflammation and fibrosis and small blood vessels. This is granulation tissue formation, right? So chronic non-specific inflammation, formation of granulation tissue, usually followed by healing by fibrosis, is chronic osteomyelitis, chronic ulcer. Now a variant of this is chronic suppurative. Suppurative mane? Suppuration mane? Ooze, pus formation. What is pus? Dead neutrophils, right? And tissue material, necrotic tissue material and dead neutrophils. Chronic suppurative inflammation in which infiltration by polymorphs and abscess formation are additional features, example actinomycosis. In fact, if there is any fungal infection, there is usually suppuration, right? Usually there are lots of necrotic areas, neutrophils. Actinomycosis is fungal infection, right? You will study this in micro. So there is chronic suppurative inflammation where along with the chronic inflammatory cells like lymphocytes and some macrophages, there are lots of neutrophils and necrosis and abscess formation, right? And chronic granulomatous inflammation is characteristic tissue response by formation of granulomas. Example, there are plenty of examples: tuberculosis, leprosy, syphilis, actinomycosis, sarcoidosis, Crohn's, right? This I've already said. Now I will, do you know what kind of necrosis this is called usually? Caseous necrosis. Caseous necrosis mane acellular formless granular material. Acellular, ওখানে কোনো সেল কিছু বোঝা যাবে না, কোনো সেলের আউটলাইন কিছু বোঝা যাবে না, সব ধ্বংস হয়ে গেছে। তো দ্যাট ইজ acellular formless cheesy pinkish material, that is caseous necrosis. So in the center there will be caseous necrosis. Along with that there will be lots of epithelioid cells. These are the epithelioid cells, right? Slipper shaped nucleus. Along with that, some of the epithelioid cells will coalesce to form giant cells. What type of giant cell is this? Horseshoe shaped arrangement of the nuclei. What giant cell is this? এটা একটু আগেই না কে বলছিল? Langhans. So there will be some giant cells and lots of epithelioid cells. Then there will be অন্য কালার দিয়ে করি। There will be all these. These are what? Color of blue color এর হবে। Color of what? Lymphocytes. এরকম অনেক, এইটুকু হবে না, অনেক হবে। Color of lymphocytes. And surrounding that, in long standing cases, there will be fibrosis. এরকম তাহলে এটা কি একটা ক্লাসিক্যাল কেজিয়েটিং গ্র্যানুলোমা। আর গ্র্যানুলেশন টিস্যুর ছবিটা আগে একটা দেখিয়েছিলাম, ওটা বোঝা গেছে। এই যখন প্রশ্ন আসবে একে টেকে ছবি করে দিলে আরো বোঝা যায় যে তোমরা আনসারটা জানো, রাইট? So granulomatous inflammation is a form of chronic inflammation characterized by collections of activated macrophages, which are the epithelioid cells, often with T lymphocytes and sometimes associated with necrosis. What happens is there is severe hypoxia at the center. So that gives rise to the caseous necrosis. It is a type 4 hypersensitivity reaction. It is a cellular attempt to contain an offending agent that is difficult to eradicate. Now, do you any of you know how many types of granulomatous inflammation are there? জানো কেউ? হ্যাঁ, টাইপস অফ জায়ান্ট সেলস জানো? Yes, foreign body giant cells. Foreign body giant cells will be found in ফরেন বডি একটা বডিতে এলো, এখন বডি কি করছে, এটা ইমিউন রিয়াকশন হচ্ছে না। ইমিউন রিয়াকশন হচ্ছে না মানে টি সেলস ওখানে ইনভলভ নেই। No T cells are involved in that kind of reaction. The body is just trying to contain that foreign body. So there is foreign body type of granuloma formation. Can be because of suture. Say a bit of suture was left. When the surgeon was removing, a bit of suture was left in that area. That can give rise to a granuloma formation. Apart from that, talc in case of intravenous drug abusers. This is also a frequently asked question in MCQs. What happens in someone is snorting cocaine? It is an adulterant, right? So talc is mixed with the cocaine. This person snorts it, it goes into the lungs. The talc gives rise to granuloma formation. So there will just be lots of macrophages, the specialized macrophages, which are the epithelioid cells. Foreign body granuloma. See, here you can see the foreign body. It is usually visible. And in a foreign body, the giant cell, the nuclei are haphazard, or a ring formation or haphazard arrangement of the nuclei in the foreign body cell, in the giant cell. That is the example of foreign body giant cell, as opposed to Langhans giant cell, where there is horseshoe arrangement of the nuclei. In a foreign body, it is either a ring or haphazard arrangement of the nuclei. So you can see lots of foreign body giant cells, this, and here there is the foreign material which is usually refractile. Form around foreign materials like talc, sutures, other fibers. It does not incite immune response. There are no T lymphocytes. Another is immune granuloma. Immune granuloma is caused by agents which produce a prolonged T cell mediated response. So here you can see, here there is no necrosis, but you can see lots of epithelioid cells. You can see this is an epithelioid cell, this epithelioid, this is epithelioid, this is epithelioid. Can you understand the epithelioid cells? Oval shaped nuclei. These are the epithelioid cells, and this is the color of lymphocytes, bluish. You can see this is more blue, this is more pink. So this is the color of lymphocytes. Now, pathogenesis of granuloma, I've already explained, this is from Hirschmohan. Now, there is cell injury, say by Mycobacterium tuberculosis. There is failure to digest agent, right? There is weak acute inflammatory response. I've already said, neutrophils agei elo, bhege gelo, kichu korte parbe. There is weak acute inflammatory response. After that, there is engulfment by macrophages. But the macrophage is not, it has engulfed, but it is not able to eradicate the pathogen. It has only eaten up. Now there is persistence of the injurious agent. So there will be T cell mediated immune response. Now this T cell mediated immune response, there will be activation of further CD4 positive means T helper cell, activation of CD4 positive T helper cells, and release of lymphokines, interleukin 1, interleukin 2, growth factors, interferon gamma, TNF alpha. And there is monocyte chemotactic factor which will attract more macrophages, and there is proliferation of T cells. The macrophages are activated by interferon gamma. There is transformation of the activated macrophages into epithelioid cells and giant cells, secretion of fibroblast proliferating cytokines like tumor growth factor beta and platelet derived growth factor PDGF, TGF beta, and there is granuloma formation. Kichu boja gelo? I have said this throughout the lecture. Same jinishta ekhane summarize kora ache. Should I repeat? So there is cell injury, attack by, just think about tuberculosis, because for all intents and purposes you will have to know tuberculosis. Baki shob auto ashe na MBBS. So tuberculosis ওটাকে কিছু করতে পারলো না, acute inflammatory cells have come, there is weak acute inflammatory cell response. That is gone. Now the macrophages have come, it is chronic inflammation, and I have said that this Mycobacterium tuberculosis incites chronic inflammatory reaction from the beginning, as opposed to say some bacteria which are causing osteomyelitis, which is what? There is acute inflammation, it is not able to contain, followed by chronic inflammation. But in case of TB, there is chronic inflammation from the beginning. So the macrophages have come, they have ingested the bacteria, but it is not able to do anything. So there is persistence of the injurious agent. Now there is T cell, now the macrophage is acting as APC, antigen presenting cell. So there is T cell mediated immune response. So there is activation of CD4 positive T helper cells. So the T helper cells are activated, and there is secretion of some chemokines, lymphokines. You have to remember, most importantly, interferon gamma, right? Because it is most important for activation of macrophages, interferon gamma, right? So there is release of some chemokines, and these chemokines are attracting further macrophages. Further macrophages are attracted. Along with that, there is proliferation of T cells. More T cells are present, more macrophages are present. Now the macrophages are activated by interferon gamma. This interferon gamma is activating the macrophages, and the macrophages is basically exercising, it is becoming larger, more powerful. So the macrophage now is converted to activated epithelioid cell and giant cells. Some epithelioid cells are now coalescing to form the giant cells. So there is transformation to epithelioid cells and giant cells. Along with that, there is secretion of some fibroblastic factors. Fibroblastic factor mane platelet derived growth factor and tumor growth factor beta, TGF beta and PDGF. These, now the fibroblastic factors are giving rise to the rim of fibrosis, and the epithelioid cells are present here at the near the center of the granuloma. The epithelioid cells and the giant cells are present. Along with that, there is the collar of lymphocytes surrounded by the periphery of rim of fibrosis. So this is how a granuloma formation takes place. Okay. কি হলো? So this is basically that. This is the central caseous necrosis, acellular pinkish necrosis. Here obviously I also can't see the epithelioid cells. I can only identify the giant cells. These are the giant cells. There is the collar of blue lymphocytes, and there is this surrounding fibrosis. This is the surrounding fibrosis. This is how it looks under the microscope. I think there are some problem cards and some slides are there with granuloma for your practicals. You will be shown all this. So last, I told you, this is like a feedback. এতক্ষণ আমি বকলাম, এখন তোমরা বকো. What is chronic inflammation? Feedback হচ্ছে 0. What is chronic inflammation? বলো সোমদীপ. Where mononuclear cell infiltration, tissue destruction, and attempts at healing are taking place simultaneously. This is the definition of chronic inflammation. Right, sit down. Cells involved in chronic inflammation. আরেকজন কেউ বলো লাস্ট বেঞ্চার, এটা তো সোজা প্রশ্ন. কেউ একজন বলো. And lymphocytes. Examples of chronic inflammatory reaction. Difference between granulation tissue and granuloma. পারবে? বলো. Yes, okay. Good. So that's it.